will be available.
Perfect. All right, so Anna is here to talk to us about infants and their onset of walking. So take it away.
Thank you so much and welcome everybody. I'm really thrilled to be here and excited to have the opportunity to give this talk. I'm here to present a work that is really a combination of skills from a very wide team. The name of the people who worked on this project are all listed in this slide. And the work I'm presenting has been published earlier this year in Nature Human Behavior. So if you want more details about the study after I have presented you, feel free to check the paper out. So the title of my talk is a Genome-Wide Association Meta-Analysis of Age at Onset of Walking. And I thought it'd be interesting for the topic of this conference because it was really merging work, effort, and data sets across different samples to really try to answer a question about the neurobiological underpinnings of an important developmental milestones. So firstly, I wanted to introduce the whole project that is led by Professor Angelica Ronald at the University of Surrey. And I started this project with her as part of my postdoc quite a few years ago. We called the project Genetics of Early Milestones and Skills. And the idea was really to gather together data sets with information about DNA, so genotype, and about early milestones and skills from infants and toddlers to try to answer an important question that is, how is genetics influencing the development of behavioral traits? And as you can see, the aim of the project was to bring people's attention on the topic, and we made the cover for Nature Human Behavior. So at least for the first of our publications, we hope we reach the goal. So as I was saying, the GEMS project aimed to address questions about what genetic influences we can measure that had an influence on early milestones and skills. And our key candidate for the beginning of this project was age at onset of walking. And why? Firstly, because it's a simple yet robust marker of brain and behavioral development, and it's used in clinical practice as well to signal how development is progressing for individual children. It's also clearly defined and reliably recalled by parents, so we kind of could trust the measure and the fact that it could be a reliable measure that we could use in our project. And also because there was a lot of attention on physical, cultural, and environmental factors that influence these important motor milestones and that couldn't explain the variability between individuals fully. And from these important studies, we know that age at onset of walking is linked to later outcome in the field of neurodevelopment research, and it can have cascading effects on other developmental domains. So for example, when a child is able to walk independently, they can decide where to go independently from their caregiver. They can reach new areas of the environment. They have their hands free to manipulate objects. They can make new experiences and learn new things. The motor milestones have been studied a lot, even from the World Health Organization, just because they are indicative of a children's development and used in clinical practice. And as you can see in the graph indicating the windows of achievement of the main gross motor milestones, age at onset of walking is on average achieved around 12 months of age, but there is a wide variability in the age at which children learn to walk independently. It can go from 8 months to 18 months, and that's still in children with a typical development. And we were asking, what are the biological mechanisms that underlie the wide variability in age at onset of walking? And also, are these biological mechanisms also associated with later health, neurodevelopmental, and cognitive outcomes, as we can imagine from studying age at onset of walking at the behavioral level and following up children at the behavioral level? We didn't have much information about the biological mechanisms involved in age at onset of walking, but a recent meta-analysis had indicated that from twin studies, we knew that heritability of psychomotor functions in general is pretty high. So twin studies usually correlate traits acquired in monozygotic twins that share 100% of their genome, with a correlation for the same trait that is observed in dizygotic twins that share only 50% of their segregating DNA in general. So if monozygotic twins are more similar on that trait than dizygotic twins, we infer that the trait is highly heritable. The genetics has a role in it. And this is the information we had at the moment of starting this study. We also had information from a twin study conducted in England and Wales, specifically on age at onset of walking, that showed high heritability. 84% of the variance between individuals, or the variability between individuals, could be explained by genetic factors. But we had a search of the literature, and there was no study that investigated the actual molecular DNA linked mechanisms underlying in this motor function. And so what exactly were we looking at when looking at the DNA? This is for those of you who are not familiar with the DNA structure. So the DNA is a double helix strand that carries information about what proteins need to be produced and when in our organisms, and it determines the development and the functioning of a human being, and it's carried inside the nucleus of our cells. It's all wrapped around proteins to be protected, but if we unwrap it, we see that it contains a sequence of bases, and almost 99.8% of the DNA is identical among human beings, because that's how the human being should be and should develop. But there are specific locations in the DNA that we have identified where I can have an A, or you can have a G, someone else can have a T. So we each can have a different nitrogenous base in that location. And that's commonly observed. The rarest of these are commonly observed in the population. So it's observed in at least 1% of the population. It's not something rare or a disruption of the DNA. It's quite normal to have a variation on these locations that we called single nucleotide polymorphisms, or SNPs. And these differences in the DNA have been linked to differences in physical but also psychological traits. So these differences are those that we were looking for and that we define common genetic variation. And the way we measure these is that we go at the level, we go to the individual and we collect cells from their body. So for example, from cheek swabs or saliva, if we want skin cells or blood cells. And then we extract the DNA, we enlarge it to be able to read it. We put it in an array that is going to color code using fluorescence based on which base is observed in the cells of that person. for those specific locations in the DNA. And so what I obtained at the end in my computer is a list of SNPs, so locations where we expect the variation and what basis the individual carries at those specific locations. And then I can pull together all the sample that is genotypes of which I have obtained this information and test for association with a phenotypic trait with multiple linear regressions for each of the SNPs that I obtained. And so this is the approach that we used in a genome-wide association study that is doing exactly this. It's testing the association at the level of the entire genome in these locations where we expect a variation. So we take DNA from different cohorts. In the case of this study, we had 4 European cohorts that participated, that collected genotype information from their children and also parent report information about the child's age at onset of walking in months. And then we conducted this genome-wide association study, testing the association between genotype and age at onset of walking in each cohort. And then we meta-analyzed the effects across the entire genome. So the total sample size for this analysis was 70,560 children. So I'm going to tell you something about each of the cohorts that participated in the study, because my aim today is really to let you appreciate how much of a collaborative effort this work was. So the largest cohort that participated in the study is called the Norwegian Mother, Father, and Child Cohort Study, or MoBa. And the participants were recruited all over Norway. And the parents were asked this question: how many months was your child when he or she took their first steps unaided? And the question was typically asked around 18 months of the child, and then again at 36 months. And what you can see here in the graph is a histogram of the distribution of this data for the MoBa cohort. The second largest cohort was called NTR, or Netherlands Twin Register, is a cohort of 6,000 twins and multiples recruited in the Netherlands, and parents were asked, with how many months could your twin walk without support for the first time when the children were around 2 years of age. The third cohort was Lifelines. It's again another cohort from the Netherlands, north of the Netherlands. For this cohort, the question was asked retrospectively. So the children, when parents answered this question, were aged from 3 to 10 years of age. And parents were asked, when did your child start walking 3 to 5 steps without help? And they had different options where months were indicated. So very early, average, late, very late, or I don't remember. Obviously, those who were answered, I don't remember, were excluded from the study. And because in the Genome-Wide Association Study, we needed a continuous variable with Professor Frank Durbridge, who is a statistician in our team, we decided that a good approach would have been to transform these categorical ordinal variables into a continuous variable, taking the midpoint of the age range that was suggested to parents and winsorizing the extreme of the results of the options. The last cohort that we used is called National Study for Health and Development, and it's a British cohort of 2,500 children who were born in March 1946. And then parents were asked with a follow-up survey, how many months old was baby when they walked several steps without support when the children were aged 2 years of age. And again, here you can see the distribution of the variable for this cohort. So overall, the samples had a similar range in terms of age at onset of walking. We excluded the outliers. The means were not completely comparable, indicating probably differences between the cohort. One of the difference that we expected is, for example, that the twin cohort had an age at onset of walking that was, as on average, later than other children. This is typically found in children. And this is also justifying our approach to conduct first a genome-wide association study at the level of individual studies to gather effects within a cultural and geographically defined cohort, and then meta-analyzing the effects. And we were pleased to see that our measure had some validity, because in the NTR cohort, a subset of the parents were not only asked to reply with the parent report questionnaires at 2 years, but also were phoned regularly by the team every 2 weeks to check exactly at what time the child started to walk. And the results were highly correlated. Also in the MoBa cohort, we had a very large correlation between results reported at 18 months and at 36 months, again, indicating that our phenotype measure was quite good. So the aims of this study was, first of all, to quantify the extent to which common genetic variants explain individual differences in age at onset of walking. I told you earlier that we knew about the twin heritability. Here we wanted to test a SNP heritability. So if we go and look at the DNA specifically, rather than just correlating twins. The second objective of the study was to test genetic correlation between age at onset of walking and other complex traits to see if the biological basis for these milestones were shared with other later cognitive and psychological traits and physical traits. We also wanted to discover which genetic loci had a major role in age at onset of walking, and whether the functional pathways were relevant for the development of the phenotype itself. And then we also had the aim to estimate the predictive accuracy of a polygenic score. I don't have time to go into the details of a polygenic score, but basically it's a sum of effects that an individual carries of genetic propensity for a trait. And so the results show that in terms of SNP-based heritability, just looking at the DNA and at common genetic variation, we can explain almost a quarter of the variability between individuals in when they acquire this milestone. And to give you some context, autism, that is considered a highly heritable trait, has an heritability that is lower than 15%. So 24% is pretty high in terms of trying to estimate SNP-based effects on a psychological trait or behavioral trait. And we could estimate that most of this heritability was due to genes that had a function in the brain and that were expressed, so produced proteins during prenatal life in the brain. When looking at genetic correlation, we saw that the genetic variants associated with walking later were also the same that had a role in decreasing the predisposition to get an ADHD diagnosis, to have a lower BMI, a higher educational attainment and cognitive performance, and more cortical folding in the brain. We identified 11 genome-wide significant hits, so loci in the DNA that had a higher effect on, largest effect on age at onset of walking. And when looking at these locations in particular, we could identify some that were in the vicinity of genes that play a role in neurodevelopment. And actually, we could merge data with a team that looks at mutations and disruptions in genes. And they identified a few syndromes where these genes were disrupted and individuals showed different difficulties in neurodevelopment. In terms of the polygenic score, we found that we could use a polygenic score to predict this trait in independent cohorts. I don't have time to explain the methods, but one of the interesting analyses that we carried on in the twin cohort, the NTR cohort, was to compare effects at a statistical level between within family and between family effects. And we didn't find a difference, a significant difference in the effect that the polygenic score prediction had, indicating that really what we were observed were direct genetic effects and not effects mediated by the genetic influences on the environment that were more similar within a family than between families. And we also tested a polygenic score in a different independent cohort, the Development Human Connectome Project cohort, trying to predict Bailey's gross motor skills at 18 months. And again, we found a significant effect there. In the same cohort, we were also lucky enough to work with the team who processed MRI data to look at the volume of brain areas at birth in the children, and we found that the polygenic score was associated with volume of several areas that are involved in motor control. So overall, we found that age at onset of walking is indeed associated with common genetic variants that operate in the brain. And we discovered that loci involved in these, genetic loci involved in influencing age at onset of walking have a plausible biological relevance to the trait. So in a way, confirming that what we were looking at was true signal that had a biological function. We also saw that age at onset of walking is partly influenced by the same genetic variants that also influence individual variability in other complex traits measured at later ages, including ADHD, educational attainment, BMI, and brainification. And we also saw that most of the genetic signal identified in age at onset of walking GWAS to produce a polygenic score capture direct genetic effects. And so it's not really confounded by the effect that genetics can have on the environment that in turn influences the phenotype. Final considerations about these analyses is that we made all the efforts to include samples that had a highly similar and reliable phenotype and that had a sample size greater than 1,000 to allow us to reduce the standard errors in the estimates of the individual effects that each of the loci could have on the phenotype, but this naturally restricted the number of samples that could be used in the analysis. Another consideration is that, as I was trying to mention earlier, the measurement was highly consistent across cohorts, and the fact that we did identify genome-wide significant loci using a very stringent threshold for correction for multiple testing of the p-value is in support of the validity of the phenotype measure that we chose to test. There is naturally a potential attrition and participation bias that needs to be accounted for all studies considering population cohorts, and especially for a study on development that included the availability of genotype and phenotype data from a developmental cohort. So this is something that we needed to consider when thinking about generalizability of the samples. And also linked to this, it's also that at the time of starting this study, only Western European cohorts were available. So our results are naturally not generalizable to children from other backgrounds. To conclude, we could find evidence that inter-individual variability in age at onset of walking is partly due to common genetic variation, but this was only possible by emerging efforts from different teams and different cohorts. Future research should make this effort even wider and explore how genetic predisposition interact with early environment known to be linked to age at onset of walking, and to expand this in diverse populations to obtain more generalizable results. So with this, I want to acknowledge Professor Jellica Ronald, who led the study, Anya Hollowell, who also conducted the study with me, and who is also focusing on other infant traits. So if you want to check out her work, which is currently under the review but pre-printed, you can check the paper on Genome-Wide Association Studies of Infant Temperament, linked to later life outcomes. And this study includes samples from non-European ancestries as well. So thank you very much for listening, and I'm open to a few questions.
Wow. I echo the other Erin's comment in the chat here that that's an incredible sample size for especially this kind of study. I don't know that I had any here. Double Erin, she says. Agree. I didn't really have any questions, but biology was a long time ago, but I think you explained what was going on.
Yeah, I know it's a lot for 25 minutes, but I hope I just conveyed the message that bringing together people expertise from different teams and areas in the world is really key to answer some key questions about neurodevelopment as well. So thank you everyone for listening. And if you have questions, you can email me. I'm happy for the team to share my email address.
I'm gonna try to sneak in with a question before our 2 minutes are up, but could you talk to me again about, I don't think I quite followed the explanation of why the samples needed to be so large from each group.
Yeah.
Because one thought that I have is like, oh, like I do eye tracking work with infants. Should I be collecting some of this so that other people who do DNA stuff can go do DNA stuff with that?
Yeah, okay, thank you so much. The idea of collecting DNA is good. Is good indeed, also from small samples, because now we have information about what genes and what loci are linked to age at onset of walking. So then now with your sample, you can calculate a polygenic score with a smaller cohort. But this is at the level of discovering which gene or which location in the genome plays a role. The reason why we need such large samples is that we're conducting a wide variety of tests. We're collecting around like thousands of tests, because we're running one regression for each of the SNPs in the genome. And
Can I interrupt real quick?
Yeah.
Sorry, that makes sense. I guess what I was wondering is, like, you had mentioned that that you were pulling from studies of at least 1,000. And so it seems like I don't know, my intuition would be the more the better. And so if you can grab another dozen studies who have samples of, I don't know, 24, or like, does that question make sense?
Yeah, but because I was telling earlier that every sample has specific characteristics, we need to conduct the Genome-Wide Association Study at the level of each sample, and then meta-analyze the effects. So that we're sure that what we're capturing is really checking the effect of a gene on a trait in a coherent, like homogeneous, culturally homogeneous sample, and not differences between cultures or like characteristics of the data collection or anything else. So yeah, the problem is that I need to run this study with a very stringent threshold for correction for each of the individual cohorts, and then meta-analyze the effects.
Interesting.
So, yeah, but now that we have information about what genes are involved, then you can use it in your smaller study with a polygeny score approach. So even with smaller samples.
Very cool. Yeah, I guess
you
I
so
didn't think about the fact that you would have to do this individually for each.
Yeah, it's just to be like to be sure about excluding confoundings, and only look at the effect of the SNP on the trait.
Okay, but on that note, I have to run the next session. So
Thank you so much,
of you go.
and thank you for the question.
Incredible work. Seems like a ton of labor went into it.